really hard, and that's how we got to know each other. And she was my little advisor, my little trajectory that took my hand and helped me through it. And I'm honored to call her a friend, and I'm honored to call her my partner, and definitely one of the brilliant minds that I've met, Avital. She has a PhD and a postdoc from NYU in cancer research, but more than that, she has a huge soul, a brilliant mind, and dedicated to really help save as many lives as we can for this disease. So thank you for joining us today, Avital, and thank you for doing this, and the floor is all yours.
Okay, thank you for having me, Tia, and I'm going to share my my screen right now with you. Can you see it?
Yeah, we can.
Okay, so I'm going to move to presentation mode. Okay, so thank you, Tia, and hello, everybody. Just before I start, I'm happy to take questions because happy to take questions. So if something is unclear or you want to ask a question, just let me know throughout. And also at the end, I'm going to leave some time for more questions as needed. Okay, my name is Avital Gaziel, as Via mentioned, and I'm heading the medical affairs team in Trajectory. And today I'm going to talk about latest innovation, or touch upon, I would say would be a better definition because it's a vast topic. Latest innovations in cancer diagnosis and therapy. So I'm going to start with asking a question. Why is it so hard to, why is it so hard to develop treatments for cancer? Why are we not being able to cure it by now? Okay, after so many resources dedicated to it and so much money dedicated to it. So what is the problem? So there are a few problems. And one thing is that is becoming clearer and clearer with more research that is being done is that cancer is extremely complicated and multifactorial disease. It's not only one disease. If we think about it, from a wider perspective. So if you look here, this is a summary almost of many of the hallmarks, or like the main, let's say, areas where cancer is actually taking over and why we're actually getting cancer. So these are like the mechanisms that are involved in the disease. So if we look here, we find some hallmarks that we know. For example, cells that just divide uncontrollably, so sustaining proliferative signaling, this thing is pretty well known. And we also have a lot of genomic instability and a lot of mutations that are accumulated in the cells. So the cells are very unstable, and every time they divide, they change. In addition, other things that are happening are these cells are resisting cell death. They are also, in order for them to sustain their growth, they need to grow a lot of blood vessels around them to get oxygen supply. So they induce a lot of what we call angiogenesis, which is the creation of new blood vessels. They activate mechanisms of invasions and metastasis, meaning that they are able to actually move around the body and populate different organs and spread. They create a lot of inflammation. They're becoming immortal, and this is something pretty interesting. Like all of our cells in the body, they're immortal, meaning that after a few divisions, like a finite known number of divisions, they would die. And cancer cells are evading this and are able to just uncontrollably divide again and again and again. And two new things that have been added to these hallmarks of cancer over the past, let's say, 5 to 10 years is the major impact that these cells have on something that we call metabolism, cancer metabolism. This is practically new, how they are deregulating the energy, the cellular energy. And one thing that I'm going to touch upon pretty in detail in this talk is how cancer cells are actually avoiding the immune system and avoiding immune recognition and destruction. And this is something that is gaining a lot of attention from the medical community, and a lot of new treatments are actually focusing these days on finding therapies that are targeting this specific hallmark of cancer. Okay, so we have a lot of different factors. So this is one reason why it's hard to get a treatment that is working. We get a lot of instability, as we said. That means that the cells keep on changing all the time, keep on mutating, which makes a therapy this is a challenge for therapy because, if your target it keeps on changing, then the therapy itself might not work, right? You get a lot of what we call resistance or refractory disease because it would work in the beginning and then it would stop working because the cells are changing, right? And then another thing that we need to remember that even if we say a person has lung cancer and another person next to them also has lung cancer, that doesn't mean that the disease is exactly the same, right? So the disease is very different can be and may be different from one person to another, which makes, doing therapy a little bit more challenging. So what do? We develop all kinds of therapies that actually try to target these hallmarks, right? So we have the usual ones, the more, the older, the classical therapies we know, chemo, radiation, that are actually targeting the more obvious pathways that we've known about cancer. But then we also have a lot of specific targeted therapies that are targeting specific alterations or changes or mutations inside the cells. We have gene therapy that is aiming sometimes to actually fix or normalize these cells. Pro-cell death compounds, drugs that restrict cellular metabolism, and immunotherapy that we're going to touch on later. So, and so on and so forth. So, so the next question would be then, how do we choose which therapy to choose, and what would be the best approach for treatment, right? Because there are so many different mechanisms, right? So many different that are going wrong. So what do we do? So in the past, when we didn't know, in the not so far past, right? We're talking about maybe even 20, 30 years ago, we didn't know much about cancer, right? Like we knew the usual things. We saw cells that are dividing uncontrollably. We know that there are a lot of mutations that are happening due to that also, or vice versa. And then we say, okay, so we are going to try and stop this division, right? So these are let's say the classical approaches. So how do we do that? We use chemotherapy that is stopping cell division. We use radiation that is actually chopping the DNA, right? And then the cells are going to die. And this is how we are going to approach it. But obviously, chemo is very established. We know how to use it. And of course, it has space, and it's still a very, very major component of cancer treatment. But with that, of course, comes because it's so non-specific, right? It has increased toxicity. And we see a lot of long-term complication, because if you are now targeting such a major thing as of cell division, then you're going to probably impact every cell in your body that is just dividing, right? So it's going to be the lining of your gut, it's going to be your hair, and then we see, of course, the side effects of chemo, diarrhea, we see hair falling off, etc., right? Because we're targeting these pathways. And long-term complications, we see a lot of heart problems, right? Weakening of a lot of muscles in the body. And of course, as usual, as I said before, these cells are extremely unstable, and we get a lot of resistance mechanisms or refractory disease. So we're not curing the cancer. We're, in a way, maybe doing what we call palliative care, right? Like we're trying to alleviate some of the symptoms, like try and postpone the recurrence of disease, or trying to postpone the progression of disease, but we're not able to cure it. And the new treatments and the new wave of treatments is starting to look a little bit different. And lucky for us, and maybe it's a good thing, that over the past few decades, we had a lot of advancement, scientifically speaking, people working furiously in the lab to try and find and understand cancer better, and understand which are the mechanisms that are so important that we need to target. And on top of that, a lot of technological advancement. So we can do things today that we could not have done even 10 years ago. Okay, and this helps us try and solve some of the issues that we have. So we have technological advances in unprecedented resolution, so we can actually get into the cells themselves and understand what's happening there. And this actually brings us to what we call a personalized approach, right? We don't do one size fits all anymore, right? Because we know that what drives tumor also differs from one person to another. So we want to approach it differently. We want to personalize the treatment, per person. And by that, we mean that we are going to base our treatment approach on the tumor characteristics, what's happening inside a person's tumor, and also personal parameters. Let's say a person's age, what they can tolerate or not, if they have some kind of what we call a comorbidity, they have a kidney problem, right? And then, you know, we have to make sure that the treatment that they're getting is actually appropriate for them. Okay, and then we ask the questions, right? If we have this resolution, we can ask which of the hallmarks is driving this specific tumor, right? Like which are the hallmarks that we talked about in the previous slide is actually relevant here. Can we identify specific players? And if yes, can we target those players? And can we also predict the response to treatment, right? Because if we have that resolution, and here we're talking specifically about genetic profiling and testing, but this could also be for protein profiling or anything molecular in the cell. So if before we look at this patient population and we look, you know, and they have the same conditions, so they are the same to us, right, in our eyes, after we do DNA profiling and we look at their DNA or proteins, etc., we can actually say, okay, so we know that out of this population, these people are actually going to be good responders for my drug, right? These would be a good candidates. These ones probably will not be able to respond to the drug because they have another mutation, let's say, that now prevents them from responding to the drug. So there is no use of giving it to them. And these people may have some side effects, right? So we might need to use some preventative care in order to make sure that they don't get these side effects, or maybe they should not get a drug, or maybe we should monitor them more frequently to see that everything is under control, right? So this gives us a lot of power. And one of the approaches that we keep on getting questions from our patients about it, it's something that is being called next generation sequencing, okay? So this is a way for us to actually be able to look at the tumor in a more comprehensive way. So with this sequencing approach, we can actually sequence a person's tumor, the whole genome of a cancer in one day, in a few hours actually. Okay, so we can look at the biggest picture that we can for that tumor. Okay, so we get a more of an umbrella view of everything that is there. It's happening very fast and it's in lower cost. And this is something that is now being introduced and implemented already into our daily routines in the clinic. We see it more and more. And, you know, I'm giving here an example from the NCCN guidelines. For those of you who do not know, the NCCN guidelines are like the Bible of the oncologist in the US, but also across the world. So it's evidence-based. By that, we mean that, you know, all the science is being gathered. We see how a therapy is being used, how effective it is, based on clinical trials, based on other real world data. And then we summarize, you know, so a doctor would come here and say, I have this patient with this profile, so what should I do with that patient? And if you can see, now molecular testing and profiling is being part of the standard of care, right? We say, in order to know how to treat that patient, you will have to look for this mutation and this mutation and this mutation first, and then we will tell you how to treat the patient based on this mutation. But this is already good news because it hasn't been the case all the time, right? We would just go from here to, okay, standard of care, you just give chemo, whatever, okay? So instead you do this step first, which is very good. But then if we look even closer, and this is the next step, and this is the future actually, is that we say NCCN guidelines panel strongly advises, broader molecular profiling with the goal of identifying rare driver mutations for which effective drugs may already be available. So we want to know the big pictures, so we can design and plan better and more optimally the management. So this is NGS, and this is molecular profiling as an innovation in cancer diagnosis and surveillance. And if we move to a more technical aspect of it, so one thing that we need to do in order to get this information is to take a sample of the tumor. okay, right? Like we cannot do any of this analysis if we don't have a sample of the tumor. And in order to get a sample of the tumor, we do something that is called a biopsy, right? We cut a piece of the tumor out, right? And then we analyze it. And this is the traditional approach, right? But this is not always feasible. There are also patients that cannot give a biopsy from their tumor because their tumor is just unapproachable, let's say by biopsy, it's too dangerous. And it's a painful and it's expensive sometimes process. And it's also not something that we do frequently. And this kind of like limits our ability to do something with the data, because we say that the tumor keeps on changing, right? So if I take a biopsy in the beginning, what does that mean for 3 months from now? So do I need to now do a biopsy again and again? And is it even feasible to actually take so many pieces of the tumor again and again? Most likely not, right? So what do we do? So another huge advancement, this is like totally, this has been around for years now, but now it's actually maturing into something that has some clinical significance, and I hope that we are going to see it in the clinic soon. It's called liquid biopsy. So instead of now taking a piece of tissue, what we do, we are actually taking a biopsy from a patient's blood. So tumor cells are always shedding small, like pieces of their DNA actually into the bloodstream. Okay, they're called cytoplasmic tumor DNA. Okay, so what you can see, here is that we can actually take these pieces of, so we can take blood from from patients, and this is an example that I took for metastatic gastric cancer. And this patient, for example, this is a test specifically for someone who has this biomarker that is called HER2. So the cancer cells are actually expressing this marker, and because of that, they are getting a therapy that is called Herceptin, okay, or trastuzumab, that is actually targeting this tumor. So it's a targeted therapy. Okay, so the patient is getting this therapy, and serially, like the doctor is collecting these plasma cells. Okay, the plasma from they're actually collecting their blood. Okay, and then we can analyze the ctDNA. Okay, and get a copy number alterations and structural variations, and we get the mutations and all this information from the patient's blood, so we don't need the tissue. And this gives us a lot of power, because now we can do a lot of things, like, for example, monitoring treatment response. So we can look at the copy number of the target and look at the tumor size and see whether our treatment is effective over time. And we can then decide to maybe stop or switch or add another therapy on top of it. Right? Because now we have like a real-time reading of what's happening within the patient's body, right? And what's happening with the therapy. Another thing that we can do is we can do risk assessment. We can actually look and see whether other molecules, other genes are being mutated, right? So let's say now that we find that, we have all of a sudden an F1 mutation that hasn't been there because it's a resistant, right? It's a change that is happening as a response to a Herceptin therapy. So if we see that, we say, okay, this patient is in higher risk of progression. They're in higher risk to actually stop responding to treatment. So we can follow them up more closely, right? Or we can decide to add another treatment or to stop the treatment, versus other mutations that are happening that are maybe helping us to understand that maybe it's better, right? Like the therapy is working better, or there is a low risk of progression, right? And then we can also design for the future, because if we know that F1 is now mutated, we can say, okay, so we are going to line up this PTK inhibitor because this would work well with this mutation. So it gives us a lot of power to understand and wisely design and manage the treatment course for a patient. And this is doable due to and thanks to this major technological advancement of introducing liquid biopsy. And this is now all over ASCO. I don't know if you know what ASCO is. I'm going to tell you in a few words. ASCO is the major oncology conference that is happening yearly in the US. This year it's all online. It's usually happening right now in June. And many of the abstracts and the innovations that are being shown this year are about liquid biopsy. So I'm really hoping to see this in the clinic and in clinical trials more frequently sooner than later. Okay, so that was the part about diagnostics and about surveillance. And now I'm gonna start talking a little bit, touch upon like some innovations in treatments. And I've decided to, to put a lot of emphasis on immunotherapy because we are in the era of immunotherapy. And I think that everyone who has been inflicted with this or knows someone or have been investigating or read a little bit about cancer lately knows there is such thing that is called immunotherapy because it's all over. And why is it? Oh, so what can immunotherapy do before we actually dive into actual innovations and actual treatments? So immunotherapy can educate the immune system to recognize and attack cancer. So a lot of times cancers, as we say, they evade the immune system from, distracting the tumor, right? They're using all kinds of masking mechanisms, a lot of different types of approaches and strategies to try and hide from the immune system. So immunotherapy can educate the immune system back to recognize and attack the cancer. It can also boost the immune cells. So sometimes the cells can see and recognize the tumors, but they're not strong enough to actually kill the tumor. So immunotherapy can actually boost this response, make it stronger. So the immune cells, the police of our body, can actually eradicate and eliminate the cancer. And another thing is that immunotherapy can provide the body with additional components to enhance the immune response. We can add on other things from the outside in order to be able to to fight the cancer. Okay, so that's all great. Why is it so promising? Why every drug company on earth is doing immunotherapy these days, and why all of our, like, not all, but a lot of our focus and resources are focusing on that? It's because there are a few things for immunotherapy. So I chose just a couple to highlight here so we understand. One thing is that immunotherapy is very precise, right? The way the immune cells are actually targeting cancer cells are very precise. Think about it, like our body keeps on, like, now with the corona, I don't think that I need to tell you all how important the immune system is. And when it gets deregulated, then we're in big trouble, right? So the immune system is extremely precise, and this is something that we actually use to our advantage when we design therapies, because that means that if we're doing it correctly, we're going to be very precise by targeting the cancer, and we're going to spare a lot of the healthy cells, right? So this would be a very, very targeted approach using the immune system. Another thing is that it's adaptive, right? Our immune system constantly changes. The cancer is changing, but also the immune system is changing, right? So together there is some kind of like a cross reactivity, you would call it, right? The cancer would change, and then the immune system would react to this and also change. So we will have better chances of avoiding what we call immune evasion, right? Like this masking or this escape from the treatment. And then one thing that is extremely important is that immunity in general is that it has memory. What do we mean by that? That if we have immunotherapy, at least we're thinking that this is the case for immunotherapy as well, that if we educate the immune system to now respond to cancer, so let's say now that we cure the cancer, we clear the cancer, if it comes back, if it progresses or recur, then the immune system is gonna kick in, and then it's going to recognize the cancer cells when they are, you don't even, the person doesn't necessarily even know that they have recurrence, and the immune system would go and eradicate the tumor, because that's what it does all the time with bacteria, right? Like we are constantly being attacked by a lot of viruses, as we know, bacteria, a lot of toxins all the time, and our immune system keeps on fighting it, and we're healthy. We're totally like, we don't know that the immune system is doing that. So this could be also the case with cancer. Like if we see it, then, you know, the immune system is like, it comes back, the immune system immediately recognizes it and eliminates it. And that actually means a chance for what we call a cure, right? Because that's what we want. We don't want the cancer to come back. And this is altogether, it's a very attractive way of fighting cancer. Okay, so immunotherapy comes in a variety of forms. We are going to explore some of them. And something that is very important to mention that maybe I forgot to mention before, this is not necessarily mutually exclusive, right? Like if we're using immunotherapy, that doesn't mean that we cannot combine it with chemotherapy, or we cannot combine it with targeted therapy or other therapies, right? The idea is that we try and target and eliminate the cancer in all kinds of strategies, and the combination strategy is very powerful, and I'll show you some of these later on. Okay, so let's start explaining some types of immunotherapies that are extremely innovative. We see some of them like in the clinic already, but a lot of them are now under clinical investigation. And by clinical investigation, I mean clinical trials. So a lot of these innovative therapies, like you can already get access to it through participation in clinical trials. Okay, so the one type of immunotherapy that I'm going to talk about is adoptive cell therapy. And what do I mean by that? Adoptive cell therapy is a treatment that uses the cells of our own immune system to eliminate cancer. So one of the cell types, the immune cell types that are more prominent, I would say, or like the strongest almost, not almost, the strongest player in the fight against cancer are the T cells, okay? There is a type of immune cell that is actually recognizing the cancer, is actually the ones that, they're actually the ones that are technically killing the cancer. So there are a lot of immune cells, other immune cells that are involved in the process, but they are the ones that are actually doing the killing at the end, okay? So there are a lot of ways for us to use these cells, okay, or to help these cells actually fight cancer. So I'm going to talk about 3 advanced approaches today. The first one is tumor infiltrating lymphocytes, or TIL therapy. Another one is engineered T cell receptor therapy, and another one is the CAR T cell therapy. That, again, this is extremely popular. I'm sure that you know, if you've been looking around, this is something that you probably heard about. I'm going to talk about it briefly. So if we talk about TIL therapy, you can see it here. So there are a lot of cases where we actually see a tumor, okay? We look at the person's, you know, tumor environment, and we see a tumor, and we see a lot of immune cells around it, but for some reason they're unable to eliminate it. They are not unable to kill it. And there are so many different reasons for that, right? So we are looking at it, and some of the reasons that we see is that these cells are not activated enough, or there are not enough of them, okay? And we are trying to see how we can help these T cells become very effective against the tumor. So they are there, they are recognizing the tumor, but they're not able to finish the job, okay? So what we do is to actually isolate these T cells, okay, from the tumor area, and then we activate them in the lab, okay? We just take them, we stimulate them in the lab, activating them, it doesn't matter how, okay? And then we are also expanding them. We make them divide, so there are a lot of them, okay? And then we just reinfuse them back into the patient. And what that does is actually boost them, okay? And help them fight more effectively against the cancer. So this is TIL therapy. It's very highly investigated in cancers like melanoma. Okay, so another approach, engineered T cell receptor. So in this case, so something that maybe I forgot to say is how T cells are actually working. So I'm going to explain about this here. So it's not always the case that the T cells are actually recognizing the tumor. A lot of the times the T cells are not recognizing even the tumor. And how do T cells actually recognize the tumor? So the more simplistic way to think about it is like a key and a lock maybe, okay? And if you look at a cancer cell, it has some locks on it. It has some locks on it, right? Like it has a certain shape on the surface of the cells. And then the T cells would come with a key. And if the key matches the lock, then the T cell would attack now the cells, okay? It would attack and kill the cells. And there are a lot of ways for the cancer cells to try and mask the lock or like use a different, mechanism to tell the T cells that even if they do have the key, this is not the right lock for it, or they cannot access the lock. So many different things that the cancer cells are doing in order to avoid this match, okay? So what we can do in these cases where the cells are not, the T cells are not recognizing the cancer cells is that we help them. And how do we do that? So we choose an optimal target. We look at the cancer, we see which locks are out there on the cancer cell, right? And then we say, okay, let's design a key that fits that lock, okay? So we take out the T cells again from the patient, and then we engineer a key, okay, as you can see here. So they already have a key, right? The green key is the key that the T cells already have, okay? And now we engineer a yellow key, okay? And then we put it in the T cells, and then we activate them. We expand them as I explained before, and then we reinfuse them into the patient. And now the tumor has the lock that fits the yellow key, and the T cells can now eradicate and kill the tumor. Okay, so this is, it may sound like science fiction, but this is actually happening. We're doing it, and it's successful. Like we have some success with this approach. The challenges is to actually find which key to use, and sometimes like how we do that and how we actually do that in the clinic, in the lab, in a successful way. So this process is actually effective, but we were seeing some promise with that. Okay, so now let's move on to talk about what CAR T is, okay, which is a chimeric antigen receptor, which is CAR T cell therapy. Okay, so it's like a more advanced and more sophisticated approach. It's pretty similar to this one, to the engineered T cell receptor, because we are again engineering a T cell, but this T cell receptor, the key that we are now designing, it's more sophisticated. The mechanism, it helps us circumvent some of the strict recognition patterns that we need. What do I mean by that? As you know, like our immune system is extremely robust, right? So we need to be very, very careful with the immune system when we're healthy, right? Because we want to protect our healthy cells from being attacked by our immune system. If that happens, there is a name for it. It's called autoimmunity, right? And there are a lot of autoimmune diseases where our immune cells are actually attacking our own body, our healthy body, right? So in order to make sure that this doesn't happen, when we are showing this lock that we're talking about, like we are showing it always in a certain context, okay? I'm not gonna get too much into the details, but context is extremely important for any key to even work, okay? The thing is that this restricts our ability to actually create these keys, right? So CAR T is, it's more sophisticated in a way that we are helping our keys to work in a more, the pattern recognition that we need, we don't need that anymore. So this is actually helping us to attack the cells more robustly, the cancer cells. Okay, and it's also helping us in a way to, so we see the same thing, right? We take the T cells, we engineer in the CAR, okay? This is the T cell receptor that is a little bit different because now it's not that restrictive in the way that it's being expressed. So it uses a different mechanism to be expressed on the cells. And then again, we expand the cells, we put them back in the patient, and then these CAR T cells are going to attack the tumor and kill it. Okay, but because we don't need this pattern recognition, this specific pattern that is needed for the key and the lock to actually work well together, we can think about almost like a development of these CAR T cells in a way that is faster, okay? Because it's like almost like off the shelves. We can design many, many keys without worrying about the context of the key anymore, right? So we can think about like designing a whole library of keys, and then this would just go into patients based on their disease, based on the expression of the locks that they have on their cancer, and we would immediately just, it will already be here, okay, and we'll just infuse them and be able to kill the cancer. Okay, so another, and I think this is, I just added it here because I feel like this is almost like now a standard of care, okay? But this is such a major part of what we call immunotherapy. So I thought of talking about it for like a couple of minutes before we move on to more innovative things. So immunomodulators. Immunomodulators are treatments that modulate the immune system. And I found this when I was preparing this, I found this really nice cartoon from Twitter. So check it out, it's really awesome. Person that is doing that. So these are just small molecules, okay? Or like in this case, they're actually, all of them are antibodies. You see here, this is the shape of an antibody. I'm gonna talk about antibodies in a few minutes in more details. And as you can see here, this is the tumor cell. And as I said, the tumor cell is somehow communicating with the T cell, and it's preventing it from working. So you see, this is tied up and the T cell is like completely like unable to do its work. And it's very sad because it cannot do anything. Even if it knows this guy is a bad guy, it needs to be eradicated, it cannot do much, right? Because of this inhibitory signal that is going on here, okay? And what these immune modulators are doing, and one of the most investigated and used in the clinic already type of immune modulator is the checkpoint inhibitors. They are coming here and they're actually disrupting this inhibitory interaction. And by doing that, they're removing this masking, right? And then the T cells are now potent again, and they're able to go and kill the tumor. So these are checkpoint inhibitors. There are many of these already in the clinic. So you probably heard of Keytruda, which is pembrolizumab, Opdivo, ipilimumab, atezolizumab, et cetera. They're all different kinds of checkpoint inhibitors. On top of that, there are other mechanisms like agonists, meaning the ones that are actually boosting the immune response to respond better to the treatment. We have adjuvants, which are compounds that are helping the innate immune response, the initial response to treatment, to be more effective. And we have cytokines that again are boosting the ability of the T cells to do a better job. So this is pretty now becoming more and more standard of care. What we are now in clinical trials, and what we see, it's the next wave. And the next wave is about combinations. So we want to see how these immunomodulators are working with different, also among themselves, right? A combination of two immunotherapies together to see how they work synergistically together, or whether we can combine them with other modalities, we call it, other different types of inhibitors, like targeted therapies that are targeting, let's say, specific mutation, or chemotherapies, okay, that are the standard of care. So we want to combine together and see how they work, and if we get even additive or even synergistic effects by combining these two modalities together. So this is what we see in clinical trials and clinical development. Okay, so I said that I was going to talk about the antibodies, and one thing that I want to mention about antibodies, and this is the more innovative parts of antibodies, because antibodies as a treatment have been here for many years, targeted antibodies. it's antibody drug conjugates, or ADCs as we call them. So in general, antibodies, they disrupt cancer cell activities, they targeted antibodies, and alert the immune system to target and eliminate the cancer. So the antibody, this is an antibody, right? It's going to be secreted from, unless we inject it. So usually they are secreted from, produced by B cells in the body or plasma cells. It doesn't matter, but we can also use them as a drug, right? Like we can design it. So this would be the antibody, and a targeted antibody would bind here to the target, to the cells, okay? And then with this part, okay, we call it the FC, it would actually call the immune system to come, okay? And then there would be an immune response there, and the cell is going to be, the cancer cell is going to be eliminated because it's being recognized by the antibody, like this, whatever marker, or in this case, it's not really necessarily a lock, as I call it, but it's another kind of pattern on the cancer cell that is being recognized by the antibody. We have drugs like that already in the clinic, as Herceptin that I mentioned before, and we have many more of these already. Okay, so this is great, but then an advancement or an innovation on that is happening right now is actually adding an anti-cancer drug to these antibodies. So since these antibodies are now targeting specific molecules on cancer cells, they're like, missiles, right? Like they're targeted missiles that are going exactly to the cancer site, right? So how does that help us? It directly delivers this toxic drug that can kill the cancer cells. So we get something that is called immune-mediated cell death, okay? So they directly delivers the toxic drug that can kill the cancer cells. And by that, it reduces a lot of the side effects that you would get. So if that's a chemotherapy, and in a lot of the cases, they're actually conjugated or attached to a chemotherapy, okay? You would get a lot of chemotherapy here. So you would get this combination of chemotherapy that is very strong and toxic for cancer cells attached to this missile that is now directing it into the, directly into the cancer site. Okay, so it reduces the side effect and is usually associated with less targeted toxic drugs. Okay, so with less toxicity. Okay, so this is how it looks at the end, antibody drug conjugates. And this is also pretty well now studied in clinical trials. So we have a lot of these, our patients have started to ask us about these kind of trials because they're very, very attractive. There are a lot of, we know of some breast cancer trials that are now using these as in combination therapies with other drugs that are pretty interesting to see how that would work actually in combination with other drugs and see the overall impact of that. Okay, so that's ADCs. Another thing that's interesting with immunotherapies is bispecific antibodies. So what do we mean by that? So if now this antibody has just have one arm and these arms are identical, this and this is exactly identical, and they're only recognizing one specific target on the cancer cell, we can actually have bispecific antibodies, meaning that the two arms of the antibodies would now recognize different things, right? So two targets at once. And if we're taking it even one step forward, in this case, we also have bispecific T-cell engagers, and you can see it here. So the antibody is actually on one arm is going to now recognize the cancer, and then on the other arm is actually going to recruit the T cells, okay? So there it's going to engage the T cells. You see, this is a bite. This is how it looks like, okay? So one part of the antibody is going to recognize the tumor, and then the other part of the antibody is going to recognize is going to recruit or engage the T cell so it can kill the tumor cells, okay? There is already one approved therapy, a BiTE therapy, that is actually approved for leukemia. But this is also something that is being investigated very heavily in clinical trials these days. Okay, so now we're going to switch gears. I'm going to talk very briefly about two more things, and then I'm going to leave room for questions that you may have. So another innovative approach to treat cancer, immunotherapeutic approach, is cancer vaccine. So this is also now becoming extremely familiar, maybe from the news and stuff. Everyone is now talking about vaccines, so you probably know more than you maybe have known a few months ago about vaccines in general. So vaccines in general, what they do is to educate the immune system about what cancer cells look like. Okay, and by doing that, we are actually able to recognize this, right? So we have all kinds of vaccines, right? We have preventative cancer vaccines, for example, like what we have for cervical cancer, and also for let's say even, you know, like vaccines, okay, that for cancers that may be caused by viruses, like cervical cancers for some, you know, for papilloma virus induced cancer, and also for hepatitis in the liver, right? So we can actually create a vaccine that is preventative, right? Because then we can prevent the development of cancer by helping the body already recognize the patterns and create immunity against it. And we also have therapeutic cancer vaccines. So this is an example of a therapeutic cancer vaccine. So we are taking advantage of the fact that there are some molecules on cancer cells that may be either expressed differently. So in this case, in prostate cancer, for example, there is one molecule that is also being expressed on regular cells, okay? But it's vastly more expressed on cancer cells. So we can, you know, think of a therapy, of a vaccine that is now recognizing this marker, this yellow marker, okay, on the cancer cells. And then, you know, when they see it, you know, like when the cancer is starting to proliferate and duplicate, and there are a lot of these, so we can actually use this vaccine to target this, and it would not affect that dramatically normal prostate cells that do not express a lot of this. But this is a little bit tricky, right? Because still this is expressed on normal cells, so we have to be careful. So another advanced approach, another advancement on this is actually doing what is called personalized neoantigen vaccines. And what do we mean by that? Because we said already that cancer cells are dividing all the time, right? And they are always changing, right? So they're creating a lot of different molecules that are specific only for the cancer cells, and they're expressing it, they're actually showing it on the cell surface. These are called neoantigens or new antigens, new markers like the yellow ones, but maybe there would be a pink one that would only appear on the cancer cell and would not appear on the normal cell. And then we can use these little peptides, okay, these little markers, to develop vaccines and educate the immune system to recognize this, okay, recognize these specific peptides, specific markers, and then we can educate the immune system to recognize it and attack it and kill it. So this is in a nutshell about vaccines. There's so much to say about it. People are doing a lifetime of careers just by developing or investigating one type of vaccine. There are many, many different types. I'm not going to get into that now. If you want to learn more, I'm happy to discuss this with you and read more about it myself in order to be able to say more. Okay, so and the last but not
we have 5 minutes, so
okay. So this is really like the end, okay? So oncolytic viruses is the last one, okay? And this is, in short, is a virus that we manipulate. We're actually inserting immune stimulating genes in a virus, and then we infect the cancer cells with the virus. We remove all the dangerous parts from the virus, right? Like all just actually create disease if we infect with that virus. And then this actually helps us destroy the cancer cells. So there is one approved for melanoma. It's called TVEC, okay, where we are actually using, I think it's a poliovirus-based oncolytic virus, where we are actually, but I'm not sure 100%, we're removing all the bad stuff of the polio. We insert stuff inside that is actually going to help the immune system recognize the cancer cells and eliminate the cancer. Okay, so how do we get access to all of these beautiful things that I just talked about? Okay, so therapies that are already approved for your specific condition. You can discuss these options with your oncologist. It's good to know, right? Like, as we always say, knowledge is power, right? So if you know about them, you can ask your oncologist about them and explore all approved options for your situation. Therapies that are approved for other cancer types, you can get access through off-label prescription by an oncologist. If they're already approved, it can be done, or you can join a clinical trial if it's available that tests the therapy for your condition. You can, if you're not suitable for a clinical trial, you can also get it through compassionate care or expanded access programs. If these drugs are not approved for anything yet, they're only strictly experimental, then the way to get access to them is by joining a clinical trial if it's available. And of course, compassionate care and expanded access programs for these drugs. And that's it. Thank you. If you have any questions, any specific questions, you want to explore clinical trials and any of the options that we discussed today or other, you can send us an email to clinicalteam@Trajectory. Our service is free for patients and caregivers, and we're very happy to help you explore these options.
Thank you, Avital, for very, very enlightening and exciting presentation, as always.
So quiet. I was like, at some point, I was like, I hope that they're there.
No. Fascinating. It was fascinating for me, as I'm sure for everybody else on the line. And I know there's a lot of people looking at us from Facebook, and I got some reach out. It was too fascinating to even stop you for a question. I know we're running out of time, but does anyone want to ask anything right now? I mean, as Avital said, she's available and can be reached out and can deep dive to anything. And we will do a series of that, and we'll deep dive for a different cancer type. But if anyone has any question on the call now, please feel free to ask. Just
Patricia is smiling and saying no, so.
I actually had one. But just because of everybody's time, I'm like waiting to see if anybody else has anything. I was wondering about the CAR-T and the TIL. So just quickly, okay. The CAR-T and the TIL and all they were very successful with the non-solid tumor, and it doesn't seem to make great breakthrough on the solid tumor for now. For a while, they were like the big promise, and I haven't seen them coming through that yet with that promise.
That's true.
More light?
Why?
Yes.
Well, I think it's more about the delivery, right? Because how, the cells, I think maybe, Patrice, you want to answer that? I think that you would be an expert too, just because I know what his PhD was about. But in general, I think it's more about like how the cancer is not only very solid, the accessibility of the treat, like the interaction also. Because non-solid are actually also immune cells. It's almost like immune cells attacking their immune cells. So it's more effective in a way, right? Because like, think about it, we are manipulating immune cells and we are attacking a different kind of cells, solid cells, right? Like different epithelial cells, right? And in the case of leukemia and lymphoma, we're actually attacking immune cells as well. So this is this is why we see some success, just because of the biology and the delivery, because it's like in the bloodstream, right? So it's easier in terms of accessibility. So that would be the reason, but we're not, I think that right now we just need to fine tune it. I don't think that it's not going to work. It's just that we need to figure out how. It's always tougher with solid tumors. Also, the models in the preclinical stage are it's more difficult when you have a solid tumor versus whether you have a tumor in the blood. Okay, so we will get there. I'm hopeful.
Me too.
Any other questions, guys? I'm happy to answer. Okay, we have one.
Yeah, we have one.
Oh, okay, I was saying I'm getting a really great presentation. Thank you. Thank you, Karin.
She's asking about the checks and balances they do in the clinical lab, putting back cells into the body. So what is the immune system will only target kill cancer cell, ensuring that the engineering on modification did not turn into those immune cells into becoming autoimmune, which is a very good question.
It's very important, right? And that we are always worried about safety, right? Because we are playing with fire almost. And this is something that is very important to understand about immunotherapy. It's always like the balance between being very impactful and effective when we are targeting the cells, like when you are targeting cancer, and not being overly aggressive so we do not attack ourselves, right? Because we need to remember, even if cancer cells are completely messed up, they're still what we call self, okay? They're still our cells. They're not bacteria, right? They're not foreign cells. So we always need to make sure that we're not overdoing it and overkilling it, right? So how we do it? A lot of safety analysis and preclinical testing before we even go to human, right? And then if we look at clinical development in the beginning of any immunotherapy, and the FDA is extremely strict about it, we see a phase 1 or sometimes even a pre-phase 1, right? Like where we are just taking a few people, right? Volunteers that, in which we test that the compound is even safe, right? We don't even touch whether it's doing anything. We just want to make sure that we're not getting this overactivation, let's say, of the immune system now. Then it's
extremely interesting and important. Any other question from the people here, or Aline, you're monitoring Facebook? Any question from there? Did you see? Not yet. So A, I want to thank you. B, I think on times of, everybody hear about the clinical trials in terms of COVID-19, and everybody understand that we need innovation on that front to get us out of where we are now. It's amazing how immensely more important it is, how immensely more complicated it is on the cancer side, but how many more approaches and innovation is coming into play, and why we as patients, we must educate ourselves and know about it in order to ask for those and be able to explore those options for us, and not to find out about it when it's too late. And I think that's our mission, and this is the role we need to be. Like anything else in life, whether we chose to be patient or caregiver or not, this is reality. So we need to take it upon ourselves, self-educate ourselves, and fight it strongly with all power. Steam ahead. Amazing, Avital. Thank you so much. So again, if anybody has any additional question, feel free to reach out to Avital. Anything you want to reach
out to us, you can just comment even on the advertisement for this talk. You can send us an email to clinical3team@Trajectory, and one of our team members would be gladly answer you and help you figure it out.
Exactly. Facebook as well, where you can reach out for there. So any challenge. So we're really looking forward to hearing from you, and we'll continue with this Trajectory innovation talks, and we'll continue bringing in more power and more information and knowledge to our community of patients. So thank you, guys. Have a great weekend, long weekend, happy Memorial weekend. Stay healthy and well.
Bye.
Everybody.